All right, AP Environmental Science. This morning we're going to cover topics 8.12 to 8.14, which deal with a topic called LD50 and another one called dose response curves. So let's kind of get into these, kind of a quick video today on these two topics. Um, so we've been talking a lot about different chemicals that can, or different substances that can get into water. Things like lead, for example, mercury, pops, all of these different types of compounds or chemicals or even elements that can get into our body and cause problems, so either diseases or some type of a toxic effect on our body. So let's just kind of review just some of the sort of general categories of some of these different types of chemicals. So we've talked about neurotoxins, right? Neurotoxins are chemicals that can affect the brain and the development of the brain. Lead is a really good example of a neurotoxin. Right? We know that lead can get into the body from contaminated water. It can get into the body through contaminated air. Right? We can breathe it in. And when that gets into our body, it can cause a myriad of problems. We could even pass that off to our offspring. Females can pass it to their offspring via breast milk. So, any type of neurotoxin that builds up in the body, we have to understand that not only can it cause problems in ourselves, but it can also cause problems for our children. Carcinogens, chemicals that can cause cancers. We've talked about a variety of different things that can get into the body and be carcinogenic. Teratogens, this is kind of a new term I haven't used yet with you. These are chemicals that can cause birth defects. So they might get into the body and may not cause any problems in the adult. But in, for example, let's say a fetus that's developing inside the mother, teratogens can cause birth defects in the unborn baby. So that can be a real issue. And then finally, endocrine disruptors. We talked about these. Again, there are a variety of different types of chemicals that can get into the body and just interfere with hormone functions. We talked about them causing difficulties in fish, for example, in the Chesapeake Bay. But again, all of these different compound or different types of chemicals, they can get into the body, a variety of different ways, through water, through air, even through contaminated food. For example, like methyl mercury with fish. They can even get into the soil and get into plants. So all of these things have their ways of getting into the human body, which again can cause problems with ourselves, but also can cause problems with children and especially unborn babies. All right, so today we're going to talk a little bit about um, just how much of these particular chemicals can actually cause damage. I mean, chances are, you've probably drank water that's contaminated with some type of a chemical. If you've ever had tuna fish in your life, chances are you've probably been exposed to methyl mercury. But that doesn't mean that you've gotten sick or you've died, obviously, or had any long term lasting effects. But to really kind of understand how dangerous a chemical can be, we need to know, what concentrations of that chemical can actually do damage to our bodies. So we're going to talk about some different types of studies and different ways we can figure this out. But this is what we call a dose response study. It says here a study that exposes organisms to different amounts of chemicals and then observes a variety of responses. We can look to see if it kills the organism, that would be mortality or lethality, or if it changes any type of behavior or reproduction or has any, let's say, non-lethal effect. We would call those sublethal effects, to see if there is, again, any type of effect on the organism. Acute studies are typically when we expose organisms for, maybe to a chemical, and then I don't know, maybe a short period of time, and then just kind of observe to see what happens. So maybe 1 to 4 days. Chronic studies, we're kind of looking at the organism for a much longer period of time. For example, they've been doing chronic studies of people from Chernobyl, for 30 years or more, just to see what the long-lasting effects of that radiation exposure was to the people that lived in that area. So that would be more of what we call a chronic study. Now, here's something that's I think this is definitely going to be on your AP test. This is called LD50, or sometimes we call it TD50. LD stands for lethal dose. TD stands for toxic dose, but it's essentially it's the same thing. The lethal dose of a chemical is basically a concentration or a certain amount of that chemical that will kill 50% of the organisms in a study. So let's say, for example, we're studying a certain pesticide, and we're seeing what its effects are on, I don't know, a certain type of insect, maybe mosquitoes or whatever it is. So if you just kind of look at the two axes, it pretty much should make some sense to you. On the x-axis here, we have obviously the dosage. We start with 0, then we move to 1. Now the units here are what we call milligrams per kilogram of body mass. So, and that's a pretty typical unit that you would see on a dose response curve, which is what this is. Then we just kind of go up, 2 milligrams per kilogram, 3, 4. Sometimes you'll see a more of a logarithmic graph, but this one just kind of goes up by increments of 1. Now on the y-axis, we have percent mortality. So this is 0% of the organisms die, 25% of them are dead, 50% are dead, et cetera, et cetera. When you get to the top of the axis, this is 100% lethality. Okay, so LD50 or TD50, what we use this to figure out is what would be what we consider to be the lethal dose. So the lethal dose is again is an amount that kills 50% of the organism. So all you have to do to find the LD50 is just find the 50% on the y-axis and then just go over to where you hit the graph and then go down. Okay, so this dosage here, 2 milligrams per kilogram, that is enough to kill 50% of the organisms in the study. Now, I guess if this was a pesticide, I guess that would be somewhat effective. But I guess if you want your pesticide to kill all the pests, you probably would want to increase that dosage, right? You probably would want to have it a little more concentrated. But this is just kind of showing us, whatever the organism is that we're trying to kill. Now, if this is, let's say, we're studying rats, how much of, I mean, this would be the same thing, right? I mean, this amount, in, let's say, a rat, that would be enough to kill 50% of the rats in the study. So it kind of the organism that you're trying to kill in this case, is kind of important, because if it's an insect, then I guess we want to have a higher dose. But if it's something like a mammal, like a rat, for example, which we're mammals as well, that amount could be really, really dangerous if it gets into the environment, obviously. This is kind of a similar graph. This is not only showing us our TD50 or the LD50, but it's also showing us something called the ED50, which is what we call the effective dose. The effective dose is the concentration where 50% of the organisms show some type of a side effect that's not lethal. So for example, if you look at the red curve, okay, if we wanted to find the TD50 or the LD50, again, go to the 50, go over, whatever, that would be, I guess, about 80 milligrams per kilogram of body weight. At least that's what it says here. All right, if we want to find the ED50, according to this graph, again, you would go from the 50 over to this blue line, and you would see that the ED50 is about 8. So that's obviously a much lower concentration than 80 is, but you can kind of just, if you just kind of look at it mathematically, the ED50, if you just take the TD50 divided by 10, you'll get the ED50. All right, so this is the concentration where 50% of the organisms die, this is the concentration where 50% of the organisms start showing some type of a side effect that's not lethal. All right, so ED50, TD50, they are two different things. Testing standards. So when we're testing, these different concentrations, one thing I think is kind of important to understand is that we can't really test every single organism on Earth. I mean, there's over 10 million different species of organism. We can't just test everything, so we have to kind of pick and choose sort of what we're testing these chemicals on. If we're looking for what we call a safe concentration of a certain chemical, like for animals, let's say rats or mice or something, then we typically just take our LD50 value and we just divide it by 10. Okay, so that's what we just did on that previous graph, right? To figure out the ED50, right? We just take our LD50 divided by 10, and whatever value that comes out to, that is what we kind of consider to be safe for most animals to be able to consume, and not really get very sick. If we're talking about humans, then we have to take this to another level, because humans, we're not rats, we're not mice. So, we basically take our LD50 and we divide it by 1,000, all right, and that would be considered a safe dose for humans. So, for example, if any chemical gets into the environment, we want that chemical to have a safe level. We want it to be underneath this value. Then we would consider it to be safe for humans, or at least safe to have in the environment. Now, when we run these tests on organisms, we typically do this in like a lab setting, and a lot of times we'll either um, use birds, sometimes we'll use fish, sometimes we'll use mammals. Typically, they use pigeons and quail. Those are the two different species that they use, because those two species are very sensitive to a lot of chemicals. So we use those animals, or at least those birds, to represent all birds. Mice and rats we typically use to represent all mammals, including human beings. So if we consider something to be a safe dose for, let's say, a rat, even though we kind of use them as sort of human, I guess, examples. Again, we still want that dosage to be much less for human beings. All right, so make sure we understand these testing standards. Here's just kind of a sample FRQ. It says an experiment is performed. You might see this on a test. It says an experiment is performed to test the toxicity of copper sulfate. It is using brine shrimp as a test organism. So 6 different concentrations of copper sulfate solution are prepared in separate petri dishes, and 100 brine shrimp are placed in each dish. After 48 hours, the number of brine shrimp that have died is counted and recorded. The results have been plotted on the graph below. There's 2 questions that they ask. Is what is the LD50 concentration for brine shrimp? And then question B, explain the meaning of the term threshold level of toxicity. You can see that term here on this graph. So just kind of looking at the results here, of this. Again, it's an LD50 graph. So again, you see on the y-axis, this again is your number of dead brine shrimp, out of 100. So we can also assume that these are percentages, of lethalities. On the x-axis here, sorry, this was the y-axis. On the x-axis here, they're showing you the 6 different concentrations that were used, and you'll notice these 6 different concentrations. This is kind of logarithmic, so they kind of go by powers of 10 instead of going up by 1. Okay, a little bit different from the previous graph, but that's okay. So this is the curve that results from that. So question A, we want to know what the LD50 concentration is. So all we have to do again is go to the 50% mark, and then go over, and then go down. All right, and whatever value that comes out to being, it looks like it might be like 0.08 maybe, or 0.07 or something, in that range. So whatever, again, that particular area is, or whatever particular point that would be your answer for your LD50. Now, as far as question B goes, they asked a question about threshold level. Like, what does that mean? By looking at this graph, the threshold level, it looks to me like this is sort of the point where before that concentration, which again in this case would be 0.001, but before that concentration, we wouldn't expect any problems. Like we wouldn't expect any lethalities. But after that concentration, this is where you start seeing changes to the curve. Then we would expect to start seeing some type of an effect on the organism, whether it be a non-lethal effect or a lethal effect. At that concentration, we would probably consider that to be, the threshold of danger. Before that, seems fine, but after that, that might be an issue. Okay, so make sure we can interpret an LD50 graph. All right, guys, that's it for me for today. Have a good day. I will see you soon.